and the chair for this session is Lisa Carnell. And just to remind everyone, after the session is done, we will have the investigator poster session as well as the wall gaming ceremony at 6 o'clock. Great, thank you. Lisa?
Good afternoon. It looks a little bit sparse in here. I think that may have something to do with the early start time. Not because of lack of interest, because radiation is a very hot topic for NASA these days. Before we get started this afternoon, we'd like to take a few minutes to recognize and honor one of our colleagues. And I'd like to invite Dr. Paul Todd to the stage to say a few remarks about Dr. Bill Atwell.
Thank you very much, Lisa, for this opportunity. Some of you may or may not be aware that Bill Atwell passed about a little over a month ago, and word went out all over the scientific community. I hope you've had time to peruse this projection up here. It says only a small number of things about Bill Atwell. I want to remind you that this morning you already heard about space radiation composition, the particles that are in it. Low-Z novel shielding materials and secondary radiation from the moon when galactic cosmic rays strike it, and any number of other radiation-related risks that were discussed this morning. I have to say that Bill, during his nearly 5 decades of space radiation work at Boeing, worked on almost all of these subjects, and a lot of what we know today relies on his contributions. Bill co-organized and co-chaired this very session over about a 5-year period, every single year, 5 years in a row. And in doing so, he made many, many friends in this organization. And that being said, Bill will be greatly missed and long remembered.
Thank you very much, Paul. So we have a really great lineup today of 4 speakers in a variety of areas touching on radiation and radiation-induced health effects. Our first speaker today, we have Janet Baulch. Dr. Janet Baulch is a project scientist at the University of California in Irvine. Her primary interest is in central nervous system effects and epigenetics, and today she's going to give you a talk about one of her areas of specialty on epigenetic effects following radiation exposure. Janet?
Hi, Lisa. Thank you for the kind introduction, and thank you to the organizers for inviting me to speak today. I'm really excited to be here. This is the first time I've attended one of the ASGSR meetings, and I'm really excited to see some of the new science that's out there. And so without further ado, I'll get started. And I think we can all agree, based on the talks we've already seen just today, but the talks and posters that we'll see throughout this meeting, that exploration of deep space is going to pose a number of risks to the health and welfare of our astronauts. And for me personally, I would like to argue that one of the biggest risks that they need to be concerned with are the effects to the central nervous system and brain function. And the real problem we have with looking at this is we know a whole lot more about low LET and terrestrial radiation exposures than we do about high LET space radiation exposures and space relevant dose rate exposures. And in the context of my work, the things that come to mind are, of course, what is the risk to the CNS of these exposures, and how might they manifest in a human population? And lastly, what are the mechanisms that underlie those effects, so that maybe we can find a way to protect the astronauts? And the real issue, as you've already heard quite a bit today, that most of our science and our experience with the astronauts to date on the space station or a shuttle mission is all happening within low Earth orbit. So these astronauts are protected by the Van Allen belts and the Earth's magnetosphere. Even for the lunar missions, to a large extent, the astronauts pass through a fairly thin region of the Van Allen belts, minimizing that exposure, and they were on the moon for a very, very short period of time, not weeks or months. And further, if we think about the transit time to Mars, and if the astronauts stay there long enough to explore the planet, the kinds of doses they're going to experience are significantly different from anything that people have been exposed to previously. And so one of the issues that we have with looking at this here on Earth is that we're very limited in the kinds of radiation that we can use for these ground-based studies. And we're fortunate to have a really nice high-tech facility at Brookhaven National Laboratory, the NSRL, where most of us go to perform our radiobiology research. The problem, though, with these exposures is they're exposed or delivered at relatively high dose rates, and it's usually either a single beam or a sequential beam exposure. So while these are very good in some respects, they're still poorly representative of a Mars mission relevant exposure. You're going to hear another talk in this session by Mike Weil, who's come up with his group with one possible technological advance that can help us solve that with regards to very, very low dose rate neutron exposures, but he'll talk about that more in a bit, I think. So just to reiterate though, even though there are some caveats to the work that we're doing, my lab and others in the NASA community have done numerous studies looking at CNS effects of radiation, and it includes lab like a team of Raber, Turker, and Amy Cronenberg, Bernie Rabin, Richard Britton, and Antonio Allen, and Susanna Rossi at UCSF, and Greg Nelson. And we're looking at a variety of different exposure paradigms, a variety of different CNS interrogations, and everybody is finding effects on the CNS following these radiation exposures. And so Dr. Lamoli also has some work on that, and that's in conjunction with Mike Weil's new facility. And then there's our work looking at the epigenetic response to these radiation exposures. And that's what I'm going to touch on for a little bit now. But first, let's put this again in the context of an astronaut and NASA relevance. And what we think at the moment is that the medical impact of these exposures may be very persistent. To date, people have looked out to 1 year post-irradiation in their mice, and they see no resolution of the cognitive deficits and no amelioration or reduction in those effects. Even if anything, they're more severe or pronounced at that delayed time. And these could result in or manifest as problems with memory and learning in our astronauts, as well as mood disorders and conditions that look much like a neurodegenerative disease such as Alzheimer's disease. So long-term effects on quality of life. Short-term impacts may happen in a mission-relevant time frame, where if there is an acute effect, we may see that the astronauts start to have issues with the ability to adapt in unexpected situations, or to make a strategic decision in an unexpected situation. There is one bright spot in all of this, though, the last part here, the last point that I have on this slide, and the work that I mentioned by Susie Rossi. And in the context of her work, she has seen that the female rodents are much less sensitive to the CNS effects of radiation than are the males. And if this holds true, then this may provide us some guidance or some changes in the way we pick our astronaut corps for a mission. Just something to keep in mind. And then in the context specifically of my work, I'm looking at epigenetic changes or epigenetic endpoints. And when I say epigenetic mechanisms, what I'm talking about is DNA methylation, as well as histone modifications and non-coding RNAs such as microRNAs. And these all work together really to affect the functional topography of the genome, how genes are expressed without there being a mutation or any change in the DNA sequence itself. And in the context of my work specifically, I'm looking at DNA methylation. I'm not sure, is this pointer working? Oh, no. Anyway, and so when we look at that on the molecular level, what I'm talking about are DNA methyltransferases, a family of enzymes that will take the methyl group from an S-adenylmethionine molecule and put it at the 5 carbon spot on the cytosine residue, giving us a 5-MC. And then also the TET family of enzymes as well. And these perform more of an oxidative reaction, where that methyl group is then converted to an OH to give us 5-methylcytosine or 5-HMC. And I'm going to say this on the most global, simple level, but 5-MC is typically found in promoter regions of genes and acts to silence gene expression, whereas the 5-HMC is more often found in gene bodies or in regulatory elements of genes, and that works as an activating mark in the genome. And if we look at this even a little bit more closely, this process that starts with the methyltransferases to 5-MC and the TETs to 5-HMC is an active progress or method of converting 5-MC to 5-HMC, which is a key player in CNS gene expression regulation. But it's also just one of the steps in the demethylation process too, that the TET enzymes continue to actively modify the base and take us to a 5-formyl and then a 5-carboxycytosine, and then through DNA repair, we get back to an unmethylated cytosine. So if we need active demethylation, this is also the pathway that we would use. But in the context of the brain, 5-HMC is an effective and important covalent modification in its own right. And in the context of my work, we're looking at some pharmacologic manipulation of this methylation process. And what we have is a drug called 5-iodotubersidine. And what it does is it inhibits adenosine kinase activity. And when we do that, we reduce AMP production, we get an increased pool of adenosine, and we perturb the methionine metabolism pathway. And when we do that, we inhibit the production of S-adenylmethionine, so we lose our methyl donors. So even if those DNA methyltransferases are active and gain the signal to upregulate methylation, there's no substrate there for them to work with. So if radiation induces DNA methylation, the cells still can't really act on that signal. And in the context of our NASA studies, what we've done is we've taken a cohort of animals to Brookhaven. We've exposed them to 20 centigrade of silicon particles. And in the context of either a protection or a mitigation strategy, we've either given the mice this drug for 4 days pre-irradiation with the 5th treatment at 30 minutes pre-irradiation, or we've done it in a mitigation strategy where they receive their first dose 30 minutes post-irradiation and the 4 subsequent days. Now, after that happens, we just leave the mice. We let them rest in the vivarium for a month before we begin our behavioral testing. So we do behavioral testing, and then we subsequently look at molecular endpoints in the brains of these animals. And we're using a series of open arena tasks to interrogate their cognitive function. And it's each of these interrogates memory, whether it be episodic, spatial, or recency. So in one case, the mice are in arena. They get used to studying two identical items. Then they're taken out of the arena and new unique items put in. A cognitively intact animal is going to want to look at that new object, not the old object. Similarly, if you take the animal and put it into the arena and it sees 4 objects that are in predictable locations that they become accustomed to seeing, then you take the animal out and move the location of a couple of those objects. They should be much more interested in the objects that have been moved to a new location, and that is indicative of cognitive function. Similarly, temporal order. They see 2 objects, they're the same for a while. We take them out, then we give them 2 new objects to explore. Then in the 3rd phase, they get 1 of each. And as with most of us, if it's something you just saw 5 minutes ago, you really shouldn't be nearly as interested in that as something you haven't seen for 15, 20 minutes. So in that case, you should want to look at the object that you haven't seen as recently. So all of these are measures of behavioral function. And then if we look at the data, these are data from those same animals. The top row are our protection studies, and the bottom row are the mitigation studies. And if you focus on the red column, you'll see that all of our irradiated animals show some degree of impairment on each of these behavioral tasks. And then the second most important column to look at is the black bar. These animals were also irradiated, but they received ITU treatment either prior to irradiation or after irradiation. So we're seeing at least partial recovery of cognitive function in these animals as a result of this 5-ITU treatment. And in some cases, it's to the point where they're indistinguishable from the control animals. And we've done this on multiple runs at the NSRL. This isn't just a one-off kind of experiment. This is just one behavioral task from a subsequent run. And you'll see again, the irradiated animals are impaired on what now is a novel place recognition test. And that 5-ITU is able to mitigate the effects of radiation when it's delivered in those days post-irradiation. So in the context of brain function, there are different regions of the brain that contribute to different parts of cognitive function. They are frequently, and in most cases, all talk to each other. And so for that reason, we're looking subsequently at hippocampal molecular changes, since it's critical and central to a lot of these cognitive mechanisms that we're looking at. So if you'll remember, again, 5-ITU is supposed to drop adenosine kinase protein levels in the irradiated brain. And so the first thing we did was immunohistochemistry to test that hypothesis. Did the drug do what we thought it should do? And you'll see again, the red bars, even by 2 hours post-irradiation, there's an increase in ADK level. And it remains that way at 1 month post-radiation, but the 5-ITU brings that back down, again, to something close to control in most cases. Not always, but it does bring it down some. And then more importantly, if our hypothesis is that DNA methylation underlies some of these impairments that we're observing, we have to look at DNA methylation, the 5-MC and the 5-HMC that I talked to you about earlier. And if you look as early as 2 hours post-radiation, that 5-MC level is just starting to come up, but by 24 hours, it's significantly elevated and remains so at 1 month post-irradiation. And again, the 5-ITU treatment brings it back down to something relative to control. And this will get a little repetitive, so I'll try and get through this quickly. But we also see increases in the 5-HMC levels, albeit they don't spike quite as early as the 5-MC. They're definitely significantly elevated though by 1 month post-exposure, and reduced by the 5-ITU. And further, and kind of surprising to me, is that when we look at the primary players in DNA methylation in the irradiated brain, or in the brain, DNMT3A is one of those, and radiation causes persistent elevations in that are also modulated by ITU, suggesting that even these epigenetic enzymes may be epigenetically regulated themselves. And we see the same for the TET1 and the TET3 that are both involved in converting 5-MC to 5-HMC, the second methylation mark that we're really interested in the brain. So I've just kind of flown through all of that, but I just want to get to the questions that are in my mind. And that is, these are global levels of DNA methylation that are measured by an antibody in the brain. But the most important question going forward, okay, so these global levels correlate with changes in behavior. Is it on a gene-specific level? And are these genes that are alternatively methylated in the irradiated brain and the irradiated brain plus ITU, are these the kinds of genes that would be responsible for cognitive impairments? And we also see changes in molecular and structural endpoints in the brain. post and presynaptic proteins and dendritic complexity and length and so on. And so all of these are gene driven. And so can we change these? Can we manipulate these by epigenetic means? And I just want to reinforce that while we've published on this, it's not an isolated phenomenon in the space radiation studies. That we also have work from Jacob Raber and Mitch Turker and Susie Rossi again, where they have also looked at our DNA methylation in the brain. And in this context, they've actually looked at some gene-specific changes. And in neurons in the irradiated brain, they do see changes in DNA methylation that may influence gene expression. And similarly, this is not limited to the CNS. There are a number of other investigators. They're looking at other organs in the body following space-relevant radiation exposures. And we see things in the mouse heart and in the hematopoietic stem cell pool. And I didn't get a chance to pull the reference, but I believe there's also a couple of papers out there looking at the lung. So this is not an epigenetic phenomena that's isolated to the CNS. It may play a role in the radiation response of the entire body. So just to give you some, this is totally separate, independent data from another pilot study that I did several years ago, but it does support the assertion that you can have gene-specific changes in methylation that drive our changes in the CNS. And in these studies, you can see there's numerous genes. They're involved closely in brain function. And you can also see that the changes in methylation for the 5MC mark and the 5HMC mark are often in the opposite direction. And so what I would hypothesize is that maybe this is taking us to a teamwork kind of thing where the 5MC goes down while the 5HMC comes up to turn a gene on, or vice versa to turn a gene off. And so, and this is my disclaimer because I've talked this whole time about DNA methylation, but I'm not saying it's the only answer. Yes, in our radiation paradigm, it was sufficient to rescue cognition, but it's probably not the only thing that plays a role in this. And we're going to need to look at not just that, but changes in histone modifications and microRNA and other RNA-mediated epigenetic mechanisms, and see which one of these really functionally play a role in the radiation response and radiation risk, and whether we can either protect or mitigate against these in concert with each other. And I've touched on all the first points, I think, pretty closely, but I think the real take-home message is to ask whether the manipulation of DNA methylation, does it represent a countermeasure or something that we can manipulate in order to protect astronauts from the acute effects of radiation exposure during the course of a deep space mission of a year, 5 years, whatever? And will it help mitigate those long-term effects once those astronauts are back here on Earth, protect them from those long-term quality of life issues that I mentioned previously? And I think that's really the part that I'm most interested in focusing on. And so I would just quickly like to acknowledge a few of the many people that have helped with this project over the year. First and foremost is Munjal Acharya, my co-PI on my NASA project, as well as some very talented students and past students such as Ali, Lauren Apodaca, Leila Alikiana, and Mina Markarin. And with that, I will stop. And I would also like to thank NASA, though, for their previous support of this project as well. So I'll stop at that point. Thank you.
Thank you for a great talk, Janet. I think we have about time for 1 or 2 questions. You can come to the microphone.
So you gave some data on the effects of X-ray induced damage or X-ray induced epigenetics, and also on high LET radiation.
Yes.
Do the patterns of methylation match and the overexpression of the genes match, or they're completely different from one another?
I don't have that information yet.
Janet, your talk indicates a lot of progress in this area. And in your closing question, what should we think about the possibility of giving 5-iodo-tubersidin to an astronaut?
Right, and I don't necessarily think this is the best pharmacologic inhibition or method. We've since started exploring a new one that has a higher tolerance and less toxicity issues. But I would suggest that something along these lines might be practical. And I'm not saying keeping astronauts on this on a daily basis for 3 years, but maybe if we work out how this time course actually works, maybe once a month or every 6 months or something, we need to reset. Or if it's just when we've got a high, an SPE or coronal mass ejection, that there may be some times where it's more or less appropriate. But I think this is kind of a sledgehammer, but I think we can find some more eloquent ways of doing a similar thing.
Let's thank Janet again for a great talk. Okay, for our next speaker, I'd like to invite Dr. Sylvain to give his talk on radiation-induced health effects. Sylvain is from NASA Ames Research Center, where he serves not only as the project scientist, he is also the project manager for GeneLab. Sylvain has a background as a physicist who actually also does biology, and he's been studying radiation-induced health effects for over 20 years. Sylvain.
Thank you. Oh, good. All right, let's dive into health effects. So you've seen this graph. I mean, this is a classic movie of the type of particles being emitted by the sun. Alpha and protons are the main particles coming from the sun, but you also have the cosmic rays from the stars coming on towards us, and the Earth, as we heard, has a magnetic field that protects us. So the graph at the bottom, sorry, I don't have a pointer, so it's an interesting graph because it was done by the New York Times for lay people, and they were trying to give you an idea of the type of dose you're going to get when you go to Mars. So the lower graph, where you see that long pink graph, this is the equivalent dose you would receive in comparison to an X-ray, chest X-ray. So about 2,600 X-ray for a Mars mission for 2.5 years. So it's a significant dose, as you can tell. Put it into the context. So I come from, spent a lot of times at the DOE low dose program, and this is the classic graph that we use at the low dose. It's a log scale. Each bar is a 10 log. So it goes from low dose to high dose. So at the very bottom, you see the regulation and guidelines. The second bar actually gives you the kind of like the background level for US. You can see it, sorry, hopefully you can follow me. Let me put a couple of circles here. This is the ISS, so it's 10 logs above. That's a typical mission, 0.1 sieverts for a typical mission. The cancer epidemiology starts about 0.1 gray. So around 0.1 gray, we are pretty convinced your risk of cancers are true. Let's go to Mars. So Mars is 10 times higher than that. And so here, now you're close to 1 sievert. 4 sieverts will kill you. 1 sievert will not kill you, but you're not gonna feel very good if you get it in one shot. And then finally, the A-bomb data are in that range. And so again, Mars versus A-bomb, similar dose for the people who got significant doses. Okay, note that this is the low dose level for DOE worker, radiation worker. So I wanted to give that context for you. Okay, now here is one issue, is that when you use the A-bomb data, the dose was given in one shot. It's an acute dose. ISS, Mars is going to be a chronic dose. And this is going to be a big issue in our work in health risk, is how do we go from acute response to chronic exposure. Okay, so there are different type of effects that we know of. And so I wanted to give a bit of an introduction of the things we know. So in red, you see what we call targeted effects. So this is like 50-year-old type of knowledge. This has been the paradigm for radiation biology. The target is the cell, the nucleus. So we've been focused on DNA forever in this field. And the reason why is because it's very easy to measure. DNA damage is proportional with the dose. Mutation is proportional to the dose. Cell death is proportional to the dose. Easy to measure, easy to reproduce. People loved it. Physicists loved it. And then the non-targeted effects are really this really annoying thing that we don't know what to do with in the modeling because it's not easy to measure, not easy to reproduce. And they tend to have a switch type of thing. So they don't show up and they suddenly show up, and then they don't really have a dose response that is linear. And typically they represent the target that is much bigger. It tends to affect the macroenvironment, the tissue itself. Okay, and I gave you a couple of lists here. As modeler, we try to take this data and try to predict something that we can measure at the population level. The things we love to model is the airborne data because this is the biggest radiation experiment done on human for a long time. 200,000 people were exposed to radiation. They've been monitored extensively. We know the risk. And the classic model is LNT. You can see they basically physicists who are pretty lazy with the biology decided to draw a line there. And biostatisticians love that too. I'm not a big fan of LNT between us. It's basically the idea is that there is no safe level of radiation. A little bit of radiation is always bad for you. There's nothing safe about radiation, which begs the question, living in a world that is radioactive, why is there such idea? So the concept of hormesis has come up strongly, which says that maybe a little bit of radiation is good for you. Anyway, typically the way we do modeling is that we go back and forth between the type of data we need and the data that we're gonna use to have a better prediction. Okay. DNA damage, DNA double strand break has been the big guy that we've been looking at for many years because it is the road to cancer. And typically DNA double strand breaks can be induced by environment, so radiation, for instance, but also lifestyle and also genetic. So your genetic can affect that. And in this room right now, if you were to take your blood, which I've done with many people, and taking DNA damage in your blood, I will tell you that every other cells of your body has 1 break right now, on average. Some people will have 2 breaks. And so it's constantly happening. Okay. Now, this is a classic paradigm. DNA damage happens, we repair the DNA, and then if it's well repaired, there's no cancer. If you misrepair, you're either gonna go to cell death, which is a good thing, or you can go to cancer cell. That's, you know, if the mutation is persistent and doesn't kill the cells. Okay, so one thing that my lab has been pushing for now 12 years, that first paper was in 2007, is the concept of DNA repair domain. So the classic paradigm before this paper was that DNA damage happens and this amazing repair machinery in the cells come and look for damage and fix it. So you fix it on the spot. We've been observing something strange. We're using physics and we're looking at the pattern of DNA damage from the energy deposition that's predicted from the physics. And the pattern we saw on the images we're getting didn't make sense. So the biology was not matching the physics. And the physics being so well defined, we assumed that we were not understanding the biology correctly. And the interpretation of our work was that basically breaks tend to come together into repair domain. So let me walk you through this work. Okay, so first I'm introducing to you a couple of things. The singles, you can have low-LET radiation or X-ray photon. They do the damage like it's like light. So it will really work as a wave and it will do damage in a very nice uniform distribution in the cells. In contrast, the high-LET particle, the cosmic rays, will actually have those tracks that look just like that. So those are breast cells, human breast, exposed to, I think it's 1 GeV per nucleon iron. That's my favorite particle. And you can see very nice tracks done at Brookhaven National Lab. We use 53BP1 and gamma H2X. Those are our 2 DNA markers. Okay, in my lab at NASA Ames, I brought from the DOE my mini X-ray, which is like a little device that allows me to expose cells and look at them at the same time. We can do live cell imaging of those guys. We're using microfluidics. And we also have something new at Ames we did at Ames. It's a small cesium-57 source where we can do chronic exposure and we can go to 0.1 milligray to 0.5 milligray per day, which basically represent what you can see on Mars, on Moon, and on the ISS. We can tune it. So it's pretty cool, but it's low-LET. It's not going to be as sophisticated as Mike Wai's work. You'll see later. Okay, with our technology, we can, my lab is really good at doing image processing. So here we develop a software that tracks cells. That's H2B GFP cells dividing. And actually I just exposed them to radiation. You didn't see it, but I know it because I know this movie pretty well. And we're gonna keep tracking those cells and we actually can create what I call cell ancestry tree. So we basically can look at the progeny of the cells and we know when we expose them to radiation. So remember 220 and 1094s, those are the idea of my cells. Those are 2 daughter cells that have been exposed to radiation. Now, I also have a 53BP1 marker on those cells. So let me put the movie. So, and that's really interesting. If you look at the lower left, that's cell number 220, and what you see is a number of damage. And what the cells does is dividing first, and then it creates 2 new daughter cells that come back with damage. And those damage are actually full blast, and they get repaired. And then the next daughter cells do not have damage. In contrast, 1094 decided to first repair. So we see this induction of damage. It repairs fully, and the 2 new daughter cells have no damage. Well, okay, maybe I'm very naive coming from the physics, but I would have assumed, and all my computer model that I developed always assumed that the cells, at least one division after a mother gives 2 daughter cells, the 2 daughter cells should be pretty synchronous. I would have never thought that 220 had decided to be so close to mitosis that it actually doesn't go through the cell cycle checkpoint and directly goes through division, comes back with a double amount of DNA damage that they repair. In contrast, 1094, which should be synchronous with it, decided to first repair. So it was probably a bit delayed in its cell cycle and then ended up going into a cell cycle checkpoint. Never published this work because it would have been very hard to explain this to the DNA repair society. So I decided to just leave it there. So it's an observation, not published. What you can do with movies is something pretty cool. That's a movie, we did registration, so we can do computational to put the cells back straight so it doesn't seem like it's moving. Even though it's moving, you can see the cells on the outside moving a lot. That cell received 3 gray, and let's do another trick. You see all these little dots, those are DNA damage. Now, if I take a maximum projection of that, I take all the time frame, project them, I get this image. And even though, you know, in some frame you had about 60 breaks, the majority of these breaks seems to be localized into very clear region of the nucleus. They are not everywhere in the nucleus. They are localized in some region of the nucleus. And this is really what I think it was the first evidence of DNA repair domain. The breaks are constrained in the nucleus. Okay, so we go to Brookhaven, that's the beamline. And with high LET particle, you can have pretty cool images. So that one is like, we did a big montage of, give you an idea. That's 1.5 millimeter by almost 1 millimeter wide. If I zoom on this, blue is DNA, DAPI, nucleus, red is gamma H2X. If I zoom on gamma H2X, you can see something very important for the field of radiation biology. It's called the Bragg peak. With high LET, the particle actually go pretty deep and then they stop suddenly. And actually, let me show you that differently. So this curve here shows you the, as a function, we put 2 8-well chamber slides. We tune the energy of the particle so they can stop at 8 centimeter. By the way, this morning we heard the first talk with this jacket that protects you against radiation. It won't protect you against that. And we tuned down the energy of this one. It still went through 8 centimeter. If I use the 1 GeV, it's gonna go through 12, 14 centimeters. So you would need a pretty big jacket to stop the cosmic rays. Just a side parenthesis here. Anyway, with this technique, I can, knowing where I am on the microscope, I can actually tell you what LET you have, the energy per unit length deposited. And we do that, we can actually see the Bragg peak here, and we can count the number of foci per unit length. So now the unit for this kind of particles is tracks per unit length. And the track per unit length, the number of foci per unit length, as a function of LET, saturates around 200 kV per micron. If you were to look at the intensity instead, you have a nice linear response. Okay, so let's zoom on the energy deposition of those tracks. So we're gonna go look at the track. So one thing that's really nice with high LET is that in one cells, you get both high LET and low LET. So along the tracks, you have this very high energy deposition. Of the track, you have something that looks more like X-ray. So let me explain that. If you look at the right, this is a reconstruction of a cubicle cells, if you want. And the track you see here, along this track, you're gonna get about 26 gray within 0.5 micron of the track. Outside the track is called the delta rays. Those are knockoff electrons that comes and deposit the energy just like X-ray. And in that cells, you only have 0.16, 0.17 gray. So in physics, we always say for cosmic rays, there is no such thing as low dose. Remember that. You don't have low dose with cosmic rays. It doesn't exist. You have low fluence. What you're gonna get is a Poisson distribution, and you will have cells with 0 traversal, cell with 1 traversal, cell with 2 traversal, and so on. Each traversal has a very high dose. It's very different from X-ray where you can really tune down and tune up the dose. Okay. So applying this, we were searching for the size of our repair domain, and then to make a long story short, using X-ray data, we could say that about a 1.55 micron domain explained the data that we've seen with X-ray. We use this parameter, and then now we're going to predict the response to high LET distribution of foci. And when we do that, we get this curve. We tried 4 different mathematical models. The repair domain is the top left, and that's the best model. So that's how we validated our repair domain, saying that this is probably the best explanation, that basically breaks stay within the same region, and they fall into one repair domain, and they are handled by the same repair machinery together. Okay. Why is it a big deal? Because we associating these factors. Right now in this room, like I said, you get 1 break, maybe 2 breaks in a cell. This is n equal 27, that's 27 double strand breaks, that's close to 1 gray, roughly. In this situation, you're gonna have a lot of DNA double strand breaks that are gonna be close by, and they're gonna be interacting with each other. And so the typical mathematics that if you textbook type of a probability for cell death is 1 minus exponential gamma n, n being the number of DSB. That is typically the model we use, but in this case, what we did is we added a new term taking into account those those interaction. And when we do that, we did a cool exercise. We first fitted those models. Gamma and A had to be fitted with something. We used survival curve, got this curve, and then did a prediction. And in the prediction, the red dots here you see is the prediction for 1 GeV particles. And what you can see is that they are indeed much more efficient at killing than the X-ray. And actually, we did that for all particles that we know in space. And we could reproduce nicely the RB that we see. So we now have a nice risk model for cell killing in space. Okay, let's move away. I don't know how much time I have left, so but I'm towards the end. Let's move away from the cells and let's move to organ response. So one thing that we've been studying a lot is breast cancer. I don't know if you know that, but the only known carcinogen for breast is ionizing radiation. Okay, so we use the mouse model, and my mentor, Marian Barcelosov, developed this great system. It's called the radiation camera model. So very briefly, those things you see here is the fat part of a breast, and that's a young animal. Those little things here that you see is the epithelial bud, and that bud actually will make a full gland. So the nipple is on the left side. And then now what we do is on the animal, we remove this before it grows a breast. So now this animal will never have a mammary gland. But then what we do is we inject back inside a tissue from another animal that is actually prone to develop tumors. And so it's another breast tissue, and it will grow. It will not always make tumors. That's the model. You need to use the right system, so it doesn't always make tumors. And what we do is the following. We expose the animal that has been cleared from the radiation, and then we inject those cells, and we see if radiation has increased the percentage of tumor or decreased. And to make a long story short, this was published in 2000 and 2011. The paper in 2000 was the great paper, but they were using a special cell that had some people were not happy about the cell they used, that they injected, so they repeated that with a P53 null cancer cell system. And again, the purple curve shows you that irradiating the stroma is sufficient to increase the propensity of cancer. So basically, you're promoting cancer. No DNA damage here. And that's really the big difference. We are basically promoting cancer by exposing the breast tissue itself, the environment of the cells. The low-dose program loved this model, and they repeated this using a back-cross system. They took the Baulch, which was the animal they used in the previous data, and they used the spread, which is a resistant animal. They don't develop breast cancer. Mix them up, create a bunch of unique animals, repeated the clearing fat pad techniques. And big surprise, after 1.5 year work over hundreds of animals, they got the opposite response. It was protecting the animal. The radiation, the exposure of the breast environment actually protected them from cancer. And that was a big surprise. We did some GWAS on that, and basically the spread was inferring some loci that has been identified to infer protection against cancer. And then the animals, the few animals that developed more cancers, actually the loci were associated with the biopsy. So basically what it tells you is genetic is a very important factor that predicts the risk at this level. So non-targeted effect seems to be very prone or very dependent to the genetic of the host. Okay, we model these kind of things by using two-stage model. Going fast here, I'm referring to the paper. We use automata, which is kind of like little entities, computer entities that we can program. And so each query is a cells, and we can tell the cells behave this way in certain condition, behave that way in other condition. We can make mutate, we can then die. And we basically use this kind of model to first predict breast cancer incidence in humans. So we first did the edge dependence, and then we actually plugged in the EBOM data, fit the model, and then applied DNA damage model that I showed you earlier to predict the cancer incidence from exposure to cosmic rays. So this work's been published, if you're interested. We also work on something else that I thought was super interesting. I don't know if you know that, but basically young women are very sensitive to radiation. And here we show that basically using modeling that if a woman is actually having her breast growing and getting exposed to X-rays, she will become like a BRCA1 patient. Her risk of breast cancer is 6 times higher than a normal person. And this is due to the fact that her stem cells are being increased by the radiation. So there's a self-renewing signal that happens, and the breast tends to be more prone to cancer. So I think I should stop here. This is if you're more interested in this work, I'm going to show tomorrow some data on the HRP grant that we got, where we're looking at GWAS with respect to human and their risk to cancer using DNA damage. And there's also more work on GeneLab as well that I can show you later. Good? Thank you. Is there any question?
We have time for 1 or 2 questions. Does anybody have a question for Sylvain? Okay, and our next speaker we have today, Dr. Mike Weil. He's a professor of cancer genetics at Colorado State University and the director of the NASA Specialized Center for Research on Radiation Carcinogenesis. He's going to talk to us today about chronic neutron radiation exposure.
So I'd like to start by thanking Lisa for the invitation to come and talk with you about the low dose rate neutron irradiator facility at Colorado State University. The topics I'm going to cover today are going to be the rationale for using low dose rate neutrons to simulate protracted space radiation exposures, tell you a little bit about a facility we've set up to do that at Colorado State, and if I have time, tell you a little bit about some of our early findings. So you've seen this before. Beyond low Earth orbit, astronauts are exposed to solar particle events, primarily protons, energies up to a couple hundred MeV, but something that can generally be shielded against. But also galactic cosmic radiation, which is another animal altogether, originating outside of the solar system. And included within galactic cosmic radiation are HZE ions, or high energy, high Z ions. These are essentially atomic nuclei stripped of their electrons and moving through space at near light speeds. And as you've heard from Sylvain, they or their fragments are capable of punching through any practical level of shielding. So we don't have very much experience with HZE ions. As has been mentioned in several talks, even astronauts in low Earth orbit are pretty well protected from them. The only real experience is with the lunar missions. And so what I'd like to show you here is a piece of Ilford G5 emulsion. This was strapped to Neil Armstrong's ankle during Apollo 11, the first moon landing. That mission lasted 8 days. And what you can see on this emulsion, which was developed when he got back, is a smattering of X-rays and gamma rays. So those are the little specks across the film. You can also see several tracks. Some of them have light densities to them. Those are protons. And then you're going to see one that cuts diagonally across. That's an HZE ion. That's probably a calcium ion. and look at the density of the ionizations around that track. So this is what we refer to as high LET, or high linear energy transfer. Now, I want to put this in perspective for you, move it to the biology. So what I'm showing you now is a photomicrograph of a section of human liver. And I've scaled it to exactly the same scale as that G5 emulsion. And so now I'll do an overlap for you. So on an 8-day mission, this is what the radiation environment within a tissue looks like. So let's talk a little bit about relevant doses and dose rates. These have been measured aboard the Mars Science Lander. And for deep space in transit, we're talking about 0.5 milligray per day. On the Martian surface, about 0.2 milligray per day. And on the lunar surface, it would be a little bit higher than that. For a Mars mission total for 600 days, we're talking about 250 to 400 milligray. That is not a low dose. It's delivered at low dose rate, but that's not a low dose, and it's going to have biological consequences. So one of those consequences is, of course, cancer, and the cancer risk is modeled. This shows you the estimated cancer risk for a 940-day design reference mission. And that first red block there is for, that's going to be for a female astronaut with a point estimate of about 3.5%, meaning a 3.5% excess risk of developing a fatal cancer as a result of that mission. And you can see that the upper confidence limit is much higher than that. For a male astronaut, it would be about just under 3% risk, but the upper confidence limit on that is about 6.5%. Now, the permissible exposure levels are for 3% at the upper 95% confidence interval. So what you'd like to do is squeeze down those confidence intervals, and for that you need to know the major sources of uncertainty. And the major sources of uncertainty in the NASA risk model are radiation quality effects. So the effects of low LET radiation versus high LET. Remember, the epidemiologic data is for low LET, predominantly from the Japanese atomic bomb survivors. And also dose rate effects. So the atomic bomb survivors were exposed to radiation instantaneously, whereas astronauts will be exposed over a very protracted time, months to years. So why is dose protraction important, particularly with regard to cancer? So here are some results with low LET radiations, and this is a mouse study. Mice are irradiated with gamma rays or X-rays. Some of them develop myeloid leukemias. And what you're seeing in the solid line is the incidence of leukemias in mice irradiated with these various doses. And you can see you get an increase up to about 3 gray, and then a slight decrease after that. If you take that dose and instead of delivering it acutely, you protract it, you get the results in that dotted line. And what you're seeing there is dose sparing. You get fewer tumors. And so that's pretty much what we go with. Okay, low dose rates equals fewer tumors. Is that really the case with high LET radiation? Well, there's epidemiologic data in humans that suggest maybe not. So humans exposed to radium-224, which is an alpha emitter, actually had more osteosarcomas if they were exposed over a protracted period than over a relatively short period to the same total doses. Also, uranium miners exposed to radon progeny, once again, you see if they're exposed to the same doses, but over a longer period of time, they're at greater risk. So that's a so-called inverse dose rate effect. Now, we also have good experimental data with rodents. And for the high LET radiation, fission spectrum neutrons are used. And so here's an example of a rodent experiment. This is done by Bob Ulrich, and it uses californium-252 neutrons. The dose rate is 10 milligray per day, or about 20 times higher than a dose rate in space. And so you're looking at mammary tumors. The upper one of those curves, that incidence for mammary tumors is from the protracted exposures. If you gave the same dose of neutrons, but instantaneously, or not instantaneously, but over a very short period, you get the lower curve. So this is the inverse dose rate effect. Now, I don't want to mislead you. I don't want you to think you're going to see this with every tumor type, but you do see it with a number of different types of tumors. So obviously, our question is, well, what happens if you drop it down to something that's more approximate to space radiation? What happens if you drop it down to 1 milligray per day? And so that's our rationale, is we want to get data at 1 milligray per day, which we think is a space-relevant dose rate. And we're interested in using neutrons for a couple of reasons. First of all, astronauts are exposed to neutrons, and those neutrons are generated by the collisions of HZE ions with the spacecraft and also with the astronauts themselves. And as we heard earlier today, albedo neutrons as well from HZE ions striking a planetary surface. So what you're looking at here are integral neutron spectra, and the dark line, the solid line, is going to be from a californium-252 source. The long dashed line is going to be the spectra for of what you would expect to see inside of a spacecraft with about 2 centimeters of aluminum shielding equivalent. And that other line, the dotted line, is going to be what you'd see for a habitat. And what you can see here is that fission spectrum neutrons nicely cover a lot of the regions, a lot of the energy regions for the exposures that's in space. But the main reason that we're interested in fission spectrum neutrons is as a model for high LET effects of HZE ions. So neutrons, when they strike tissues and all, hit atomic nuclei, cause them to recoil, primarily protons, and that transfers energy. And so you have various LETs here, and I don't have a pointer, but if you look at the fourth bar, that's going to be the LET, the dose average LET for neutron recoils. The following bar is what you would see for a typical spacecraft, and the following bar for a typical habitat. For Sylvain, his favorite ion is iron at 1 GeV, and that's the first bar. So we're in the correct LET range with fission spectrum neutrons to be relevant for space exposures. So how do we actually do this? Well, this is the CSU Foothills campus, the entrance to it. And sitting on the campus are 3 of these structures we call blockhouses. These were built in the 1960s to do radiation studies using cesium, so low LET radiations. Those blockhouses were decommissioned in the early 1970s. We got a hold of one in order to modify for neutron use. And I'll point out, my colleague, Dr. Borak, refers to that area as being a pastoral setting. You cannot afford to leave dirty mouse cages or biological waste out on the loading dock at night. It will be attacked. We have a picture of the culprit. We haven't identified her yet because she's wearing a mask of some sort. We also have mountain lions from time to time. So this is an email telling us, be careful, and rattlesnakes, of course. So anyway, we get a hold of one of these blockhouses. We clear it out. We put in an industrial strength HVAC system and a really good humidifier, and go through a bunch of regulatory compliance stuff and paperwork, and install in the center of it that blue thing, which is a neutron irradiator containing a californium-252 source. The yellow thing that you see wrapped around it is an attenuator made out of lead to cut down on gamma ray contamination. Californium-252, besides giving off neutrons, also gives off gamma rays, and we wanted to keep that down. You can see that the irradiator is surrounded by mouse and rat caging. And when the facility is full, which it had been for a while, we have up to 900 mice and 60 rats being irradiated continuously. So Dr. Borak, who's in the audience somewhere, takes a look at the dosimetry here. And what you're looking at here is the dose that's contributed by gamma rays. So that is the dashed line at the bottom by lineal energy. And then you also see neutrons. And so about 80% of the dose is coming from neutrons and the other 20% from gamma rays. How does this compare with what you might see with space radiation? Well, space radiation is going to be that orange block. And what you're looking at is flux or fluence. And this is by LET. So LET is on your x-axis. So what you'd actually have in space looks something like that orange block. If you want to get the best mimic for that here on Earth, you go to the NASA Space Radiation Laboratory, where they use a multi-ion cocktail of sequential beams, and that's going to be that reddish purplish color. If you look at fission spectrum neutrons, you get what's underneath that black curve. So once again, we're in the appropriate range. If you're interested in the dosimetry, paper has just come out by Dr. Borak describing the facility and the dosimetry in detail. So we've had a number of different groups that have done work out at the facility. Obviously, you want to get as much use out of it as possible. So we've had groups that have looked at carcinogenesis, neurobehavioral effects, countermeasures, cardiovascular effects, CNS effects. And in addition to that, one group, for reasons that kind of elude me, looked at in utero effects. We provide them with support services, so we help them with their animal care and use protocols, getting them approved. If they want to come on site, we get them access to our network. We can provide them with veterinary residents to do some of the work, or student hourlies if the work is not very sophisticated. Help them out with tissue collection and processing. And we have equipment and procedure space that they can put instrumentation. So for instance, behavioral testing equipment. This facility is only a few hundred yards away. It's a regional innovation center, and we have procedure rooms in there that outside investigators can use. So in terms of the status and availability, the way that this actually works, when I refer to a milligray per day, is that we leave the source exposed for 16 hours per day, so the mice are continuously irradiated. Then we retract the source into a safe position, and that allows us to do husbandry, check on the animals, do facility repairs, that sort of thing. And then we start our next day after that. Well, Californium-252 has a half-life of 2 years. So what you're doing is every day you're bumping up the time a little bit. And so by the time you get out to 400 days, you're going for 21 hours, and that leaves you with 3 hours of husbandry time for 900 mice and 60 rats. At that point, it's time to replace the source or lower the dose rate. So we've depopulated the facility and we're replacing the source, and that new source will be back in operation and the dosimetry done. We're planning for summer of 2020. So I'm interested in carcinogenesis, and we have our first batch of animals that came through in a carcinogenesis study. And this particular group was Baulch female mice. It takes a long time for them to develop cancer. So we've only recently gotten them all the way through their lifespan, which is 800 days. We terminate the experiment at 800 days. So at about 100 days of age, they go into the irradiator. They're exposed for 118 days, 200 days, or 400 days to the doses that you see. And at this point, we don't have histopathology, so I can just show you life shortening. So lifespan shortening. And what you can see is that you get lifespan shortening as a result of this. And the shortening at 0.4 gray is, or 400 milligray, is about 10%. So if you want to put that in perspective, these are female mice, so we'll take a female astronaut, and I've gone to actuarial tables and asked the question, 45-year-old, college-educated, white female in very good physical shape, drinks 1 to 5 drinks a day, or pardon me, a week. I wonder how I made that slip. Okay. And asked the question, if she wanted to shorten her lifespan by 10% and she didn't want to go into space and get 400 milligray of high LET radiation, what could she do instead? And the answer is she can smoke half a pack to a pack of cigarettes every day. Okay, so we've also looked at cardiovascular effects. This is work by Mariam Burma and Adam Chicot. And what we're seeing with echocardiography is an increase in left ventricular posterior wall thickness, and that corresponds to a decrease in function. Okay, and this is backed up with mitochondrial data as well. And I'm going to skip in utero exposures. Anybody who's curious, I can answer about that later. And I'd like to acknowledge the work of Tom Borak, who's our physicist on the project, Lawrence Halbrin and Adam Russick, who came and checked out the facility to make sure everything was correct, and funding from NASA and also from Colorado State University to get the facility up and running. And with that, thank you very much. Appreciate your attention.
What do you charge for using the facility? What's the pricing structure?
Actually, the way that it works kind of depends on how it's set up, whether it's set up as a subcontract on a grant or just fee for service. So if it's set up for fee for service, you're going to be paying about $1.10 a day for a cage of mice in the irradiator, $1 a day for the irradiator use per cage. And that's really about it.
So when translating these effects in the mouse to a human, in my experience with other disease areas, the spinal cord injury, we'd have to cut a nerve, for example, completely cut out the thoracolumbar region, and over a certain amount of time, they would actually gain back function and regenerate. So do you see the same for mice? Mice are pretty resistant to a lot of the insults that we give them. And how does that correlate to what we might see in human? Is it really translatable?
So I suppose that may be endpoint specific. After they accumulate the dose, we know from other studies that the cancer risk remains elevated. It doesn't go away. And we're seeing most of these cancers very late in life, which is exactly what you would expect for a radiogenic cancer.
Before you go off the stage here, Mike, I have a quick question for you, which I think might be of interest to a lot of the folks here in the audience. So for the Neutron facility, right now you have predominantly mice and rats in cages there. Are there opportunities to actually use something like cells being cultured in simulated microgravity bioreactors?
So the answer to that is yes. This wouldn't be the best facility to do that in, but we have on the drawing board a different facility which will bump up the dose rate a little bit. So we already have low dose rate cell culture facilities at Colorado State, but it's with gamma rays. We're looking at producing one that'll allow us to use neutrons. And the reason for bumping up the dose rate is going to be that you can keep mice going in there for a year and a half, no problem at all. But for cell cultures, say organ on a chip type of things, the lifespan of that culture is going to be about 30 days. So you really want to get the dose in that period of time, which means that you're going to need the higher dose rate. So that's something we'd like to get online.
Great. Thanks again, Mike. And our last speaker for the day, we have Dr. Rupak Pathak he's an assistant professor at the University of Arkansas for Medical Sciences. He spent several years studying radiation-induced health effects using high LET or high linear energy transfer radiation. And today he's going to talk to us about combined low-dose radiation plus Hind-Limb unloading research.
I would first like to thank Dr. Karnal for giving me this opportunity to speak about our new low-dose radiation facility that we have developed in University of Arkansas for Medical Sciences. So this is a gamma radiation source, and it can deliver 0.06 gray per day. And we have done also some experiment using this chronic irradiation facility, and we looked at the change in the immune cell phenotype in primary and secondary lymphoid organ. So as we all know about the NASA's future plan, that NASA is interested to go to the deep space mission, and in case of deep space mission, astronauts will cross the low Earth orbit, and if they cross the low Earth orbit, then they will face various types of risk factors. And in Moon, as we all know, it is almost 200,000 miles away from Earth, and in 2020s, NASA is planning, and specifically in 2024, NASA is planning to go over there. And in case of Mars mission, Mars is about 35 million miles away from the Earth, and that plan is in 2030s. And in this case, the astronaut will be exposed to various types of risk factors. And I have taken this from the NASA website. So among various risk factors, space radiation is very important. So during deep space mission, astronauts will be continuously exposed to very low dose and very low dose rate of radiation. And these radiation are particle radiation, mainly composed of proton and helium ions and some high LET charged particles. And these radiation mainly comes from cosmic rays and solar flares, and they have higher biological effectiveness. That means they are more damaging than the radiation that we encounter on the Earth's surface, such as X-rays and gamma rays. And in addition to that, isolation and confinement is also another major risk factor, and that can induce the stress. And in addition to that, sleep loss, circadian rhythm dysregulation, and work overload will compound the issue. Distance from Earth is also contribute in this aspect. And as I mentioned, it is 35 million miles away, and this mission will take at least 3 years to complete. And astronauts will also be exposed to altered gravitational force, like on the Mars surface, they will experience 3/8 of the Earth gravitational pull, and they will also experience 6 months of total weightlessness. And hostile environment inside the spacecraft, including noise, temperature, pressure, lighting, and quantity of space are also contribute in this aspect. And based on these risk factors, NASA, and this can actually affect various cell types in our body, and it can affect the physiological functions. And based on that, NASA listed these risk factors. And I know the font size is very small, but I know in the audience, most of you are aware of this table, but I would like to draw your attention in a couple of things. So first is altered immune response. So NASA has identified that during this deep space mission, that immune response will be altered. And not only that, host microorganism interaction will be affected, and that would also influence the immune system. And from the radiation, exposure, cancer risk will be enhanced. And that would also contribute, and this is also, we know that it is also regulated by the immune cells. And in our body, thymus is the primary lymphoid organ. And what happened that stem cells from the bone marrow enters into the thymus, and they enter, the bone marrow stem cell enter as a double negative cells. So it means they are negative for CD4 plus, CD4 and CD8. And then they eventually go through the sequential developmental process, and they transformed into double positive cells. And these double positive cells within the thymus cortex transformed into single positive cells, single positive for CD4 and single positive for CD8. And these CD4 and CD8 cells, they have different functions. And these cells, the CD4 and CD8 positive cells eventually enter into the medullary region of the thymus and eventually leaves the thymus and enter into the systemic circulation. And from the systemic circulation, ultimately it enters into the lymphoid, secondary lymphoid organ, such as a spleen. Now, it has been found that various toxic challenges and even normal physiological conditions like aging can affect the immune cell phenotype and immune cell functional activity in the thymus. These are the some examples. So it has been found that chronic immobilization stress can alter the thymic immune cell population. Acute exposure to herbicide or cadmium exposure can also alter the immune cell population present in the thymus. And aging also decreases the population of CD8 positive T cells and CD4 positive T cells in the thymus. As I said, that after once it is developed, after the selection, these thymic T cells are released in the systemic circulation, and from the systemic circulation, it enters into the spleen. And in the spleen, if we look at the structure of the spleen, spleen has a distinct region such as red pulp and white pulp. In the red pulp, as the name suggests, so it is full of red blood cells, and it also has macrophages. And white pulp contains the T cells and B cells. And red pulp and white pulps are segregated by a marginal zone, and marginal zone also has macrophages. And all these immune cells, they exert differential function. For example, red pulp macrophage are mainly responsible for the phagocytosis of RBC, and they also maintain the homeostasis of iron metabolism. On the other hand, marginal macrophages, they are mainly responsible for removing bacteria and viruses. Follicular B cells, they participate mainly in T cell dependent immune response, while CD4 cells, they play important role in uptake of dying cells. So you can see that these different immune cell has specific function. So if somehow this immune cell population is altered, we can expect that that would affect our immune response. And this slide is showing that in mouse strain, that immune cell population is kind of different. It depends on the mouse strain as well as sex. So I don't have pointer, but if you look into the look into the purple color, in black 6 mice, the CD4 population is only 18%, while in 1 to 9 mice, the CD4 cell percentage is almost 50%. So you can see that different types of immune cells can vary depending on the mouse strain, as well as it varies depending on the sex. And like thymus, in spleen also, toxic challenge or physiological process like aging can alter the immune cell population. These are some examples, such as tumor-bearing Baulch mice. It has increased number of splenic CD4 positive T cells, and Plasmodium infection has shown that decreases the percentage of CD4 cells and CD8 cells, and dexamethasone treatment also altered immune cell population, and aging also decreases splenic CD4 cell percentage in the black 6 mice. Again, what I'm trying to say that toxic challenge, pathological condition, or normal physiological condition like aging can alter the immune cell population in secondary lymphoid organ, and that can alter our immune response. Now the question is, what happened after space factor? Whether immune cell percentage alters after space factor when they exposed to space factor. So here is one example that this study was done in 2003, where they have shown that hind-limb unloading, as we all know, that it is a NASA-approved rodent simulated microgravity model. And after hind-limb unloading, you can see that both the CD4 positive and CD8 positive T cell number decreases, and that decrease was directly proportional to the duration of hind limb unloading. Another study also showed that hind limb unloading causes organ atrophy, both spleen and thymus, and it also increases the apoptosis, and it also altered the immune cell population. So from these two studies, this is very clear that hind limb unloading or simulated microgravity can alter immune cell population in both the primary lymphoid organ as well as in the secondary lymphoid organ. Now the question is, if we add chronic irradiation to that, what will happen, right? But before going to that, there are some spaceflight experiment on animal studies that have shown that in animals also that thymocyte, the functional activity of thymic cell and splenic cell alters, and their population also alters, and these are the examples. I'm not going through it. And in human spaceflight mission also showed that thymic and the immune cell functional activity and immune cell population also changes after space mission. But in this case, we have to remember we can only collect the blood, right? We cannot check what happened in the spleen, whether spleen immune cell population has changed or not. Especially after deep space mission, we don't know what will happen, or whether thymic cell immune cell population has changed, or their functional activities has been changed or not. So that we have no knowledge about that. So for that, we need to have some ground-based model where we can simulate both the conditions. So for that, we developed this chronic irradiation facility at UAMS, University of Arkansas for Medical Sciences. And this facility we have developed in our Cancer Institute basement. And so before we start, we have to first make sure that no beam is escape this room. And the problem is for this room, as I said, that this this was actually a X-ray irradiation room, and this room, just above this room, there is a cafeteria. So a lot of people comes every day. So we have to make sure that no radiation goes straight up the ceiling. And for that, we first designed this irradiation facility, and we worked with JL Shepard very closely. And what we did, we put a sky shield on top of the irradiator so that beam is deflected to the side wall or in the floor, not to the ceiling. And the side walls are almost 36 inches thick concrete, and so that would prevent escaping of any radiation. And this 60-degree angle is showing the beam path. And the whole idea is to place our animal around this beam source. So this is the schematic diagram of our source. So first we designed the table that we are planning to place around the around our source, and this schematic diagram below is showing the dimension of each table, and this graph is showing the correlation between the cumulative dose and the distance. And we have found about 100 centimeter or 1 meter distance, the beam radiation fluctuation is very less. So we decided to place our animal at least 1 meter away from the source so that they get uniform radiation dose. And we first designed this plastic table. We worked with a local plastic company, and we didn't use any metal in this table because radiation may, if we use metal, then secondary electron may be produced during chronic irradiation. So to avoid that, we made this plastic table. And the upper right hand image is a image of a cage. It is a rat cage, and we made this trapezoid. We made this structure trapezoid so that radiation should be uniform. And this and we also designed the smaller mouse cage. And for this rat cage, the floor area is 175 centimeters square. And for the mouse cage, the floor area is 75 centimeters square, which is more than enough for the single animal housing as per IACUC regulation. And the lower left panel is showing our beam source. And as you can see, this beam source is guarded by 3 steel plates. Actually, we have 3 different attenuators, and we can increase the radiation dose 10 times by removing all the 3 attenuators. And the lower right panel is showing our actual experiment, and we can place 6 tables, and that can hold, each table can hold 5 rat cages and 8 mouse cages. So you can at a time irradiate 30 rats or 48 mice at a time. And we delivered 0.5 gray over the period of 1 month. 1 minute, 2 minutes, okay. Over the period of 1 month, keeping in mind that the Mars mission will be 3 years, so mouse 1 month is equal to human 3 years lifespan. And 0.5 gray during Mars mission, it is expected that astronauts will receive at least 0.5 gray radiation. So that's why we gave the 0.5 gray radiation. And this is the gating strategy to analyze the different immune cell phenotype. So I would like to just explain briefly about this slide. So this is our data, and the blue box on the right side is representing the chronic irradiation plus microgravity, and the red circle on right side is representing only simulated microgravity, and the blue box on the left side is representing the chronic irradiation. So you can see there is a trend of suppression, at least these 3 types of immune cells, Treg, CD4, and NK cells, and microgravity and chronic irradiation is showing some synergistic suppressive effect on these 3 cell types. On the other hand, in the spleen, the effect was completely opposite. And we also keep one set of mice where we expose mice just 0.5 gray one time, and we didn't see much difference. And we didn't see much difference between body weight, food consumption, and water intake. And these are the summaries of my talk. And I would like to acknowledge my lab members, intramural funding, and funding agency. Thank you so much for your patience.
I think we're going to have to reserve questions for everyone to speak with Rupak and the rest of the session speakers out in the hall. And again, I'd like to thank all of our speakers for an excellent session.
Well, I'd like to thank all of you for coming to today's first day of the ASGSR meeting. And I would like to thank all of our speakers for the day. Now, if you'll join us for the welcome reception, which starts at 6 o'clock, and also the PI poster session. So we'll see you tomorrow morning when we start up again at 8 o'clock. Thank you, everyone.